In the United States, the current lifetime risk for developing any cancer is approximately 38 to 40%. As such, chances are each of us probably knows someone who has been diagnosed or treated in the recent past. Once the diagnosis is made, a number of treatment options have to be considered, including lifestyle changes, surgery, chemotherapy, and radiation therapy, or various combinations of the above. Via the internet, we have access to abundant information regarding traditional and alternative cancer therapies. Unfortunately, it can often be difficult and confusing to determine which information is based on hard science with proven efficacy versus the anecdotal reports of someone's uncle's second cousin who claims he cured his cancer with a homemade concoction he mixed up in his bathtub. While the process of cell growth and cancer formation is multifactorial and complex, over the next few minutes, we're going to present an admittedly incomplete and overly simplified explanation of our contemporary understanding of cancer formation and growth. We'll then use that information to explain some of the different classes of cancer drugs and how diet may actually affect our cancer risk. In high school biology, we learn about the cell, the basic building blocks of all the plants and animals on Earth. Consisting of a cell membrane which holds the liquid cytoplasm and the chromosome containing the central nucleus, the cell and genetic material are capable of replication through mitosis. Producing two genetically identical cells to the parent, these cell clusters make up all the organs and tissues in our bodies, including the liver, pancreas, GI tract, GU tract, muscles and bones, and even our brains. While each of us is expected to live about 80 years here on Earth, the majority of the 50 to 75 trillion cells that we're made of won't last nearly that long. For instance, the cells that line our bowel last about 2 to 4 days, our skin cells 2 to 3 weeks, and red blood cells 4 months. The neurons in our brains and the cells of the lens of our eyes are the few tissues that don't turn over and stay with us from birth to death. For the rest of the body, programmed cell death and replacement occurs millions of times every second of our lives. As the cells of our bodies age, they can become weakened or damaged. To protect us from potentially cancer-causing DNA mutations, these cells lose their ability to divide in a process known as cellular senescence. While they may stick around for a little bit and still function, incapable of reproduction, they eventually succumb to their programmed cell death, also known as apoptosis. Through mitosis, the gap created from apoptosis is filled in with a healthy new cell. To maintain healthy functional tissues and organs throughout the body, the whole process of cellular senescence, apoptosis, and cellular reproduction is tightly regulated through somewhat poorly understood various chemical and local environmental signals. Occasionally, possibly through random genetic or environmentally induced mutations, the new cell produced through mitosis contains DNA material that turns the new cell cancerous. Fortunately, the body has a way to deal with these potentially rogue cells, most notably through this guy, aptly named the killer T cell. Part of the white blood cell or lymphocyte lineage, with the assistance of helper T cells, our immune system constantly scours the body seeking out abnormal cells. Once found, the killer T cell eliminates the threat and the tissue is replaced through the normal mitotic process from surrounding healthy cells. Now, our immune system can only do its job if it can recognize the tumor cell as a threat. To protect us from our own killer T cells, the immune system develops a sense of self, trained to bring no harm to the functioning normal cells of the body. However, sometimes these tumor cells can disguise themselves and trick the immune system into thinking they actually belong. Eliminating one of the first lines of defense, these tumor cells are then free to replicate and form other cancer cells. In addition, these genetically mutated cells may not be bound to the highly regulated process of cellular aging, senescence, apoptosis, and replacement, and therefore can divide randomly and rapidly, forming a tumoral mass in their organ of origin. The increased metabolic activity of these rapidly dividing tumoral cells requires a lot of energy, oftentimes more than what can be provided by the normal regional blood supply. In fact, The early avascular tumor could probably grow about 1 to 2 millimeters before the metabolic demands of the lesion exceed local supply and tumor cells start dying off. To maintain survival of its cell colony and promote growth, the tumor can recruit its own dedicated blood supply through a process called tumoral angiogenesis. Once a small cluster of tumor cells are established, they produce a family of related proteins called vascular endothelial growth factor, or VEGF, which regulate the growth and differentiation of multiple components of the vascular system, including blood vessels and lymphatics. Once the dedicated blood supply is established, the high energy demands of the mass are ensured and allow unimpeded tumoral growth. Okay, now that we have a little info about our contemporary understanding of tumoral development and growth, let's see how we can use that information to both diagnose and treat cancers. Let's start with diagnosis. The increased metabolic activity of a lot of cancers is a key feature of tumor cells that can help us distinguish them from the normal surrounding tissues. Using a device called a PET-CT scanner, we can actually see and document this increased metabolic activity in the tumor cells. The preferred energy source for tumors is glucose, the basic carbohydrate consisting of a hexagonal carbon-oxygen backbone with 5 hydroxyl side chains. If we substitute one of the hydroxyl side chains with a radioactive fluorine atom called F18, we produce fluorodeoxyglucose, also known as FDG, or more appropriately 18-fluorine FDG. The molecule still functions like glucose in the body and is therefore known as a glucose analog. However, the radioactive fluorine atom emits a positron, or a positively charged electron. The positron is antimatter and therefore quickly collides with a nearby negatively charged electron, annihilating both particles and producing two 511 kiloelectron-volt gamma rays, which travel away from each other at 180 degrees. The PET, or positron emission tomography scanner, can detect these gamma rays and determine where they came from in the body. Tissues that have an increased metabolic activity and use a lot of glucose, like the brain and tumor cells, are going to show up bright on the PET image. So that's a quick example of how we can use the metabolic characteristics of cancer cells to find tumors in the body. Now let's look at our various treatment options. Most of us are familiar with the term chemotherapy or chemical therapy. These are drugs designed to kill cells, mainly dividing cells. Since some cancer cells divide at a faster rate than the normal cells in the body, these chemicals preferentially destroy the cancer, but can also damage some of the normal tissues in the body, especially those that rapidly turn over, like the lining of the GI tract and the hair cells. This can explain some of the side effects of these agents, such as hair loss and nausea. There are over 100 types of chemotherapeutic agents, with some of the most familiar including cyclophosphamide, 5-FU, adriamycin, bleomycin, doxorubicin, and paclitaxel. In the mid-1900s, during the early days of cancer therapy, chemotherapy and steroids were really the only viable treatment options available. However, the intervening 7 decades have been witness to amazing advances in cancer treatment, with current therapies incorporating cell-specific, multi-tiered regimens that can include chemotherapy, surgery, percutaneous ablation, targeted therapy, hormonal therapy, radiation therapy, and immunotherapy, or any combination of the above. We've already discussed chemotherapy, so, starting with surgery, we'll briefly describe some of these other treatment options. Early researchers realized that the efficacy of chemotherapy significantly improved with less tumor burden, hence the creation of the field of oncologic surgery, subspecialty-trained physicians with expertise in cancer surgery. In the appropriate clinical setting, the surgeon can remove the main tumor prior to chemo or other therapies to increase the chance for a cure. Similarly, minimally invasive techniques can be used to debulk or eliminate the main tumor. Using image guidance with fluoroscopy, the CT scanner, or ultrasound, a special needle can be inserted into the tumor itself. Depending on the needle type, the tumor can be killed with either extreme heat or cold. Radiofrequency and microwave energy can be deposited through the needle tip into the tumor tissues to basically cook and destroy the tumor. Alternatively, A cryoablation needle uses a contained expanding compressed gas to form an ice ball at the needle tip, essentially freezing the tumor to death. The next category of cancer treatments are referred to as targeted therapies because they affect specific molecular targets involved in cancer cell growth. Broad categories of targeted therapies include signal transduction inhibitors, gene expression modulators, apoptosis inducers, and angiogenesis inhibitors. All the trillions of cells in our body possess chromosomes, 46 in total, 23 from mom and 23 from dad. Contained within these chromosomes are the double helix strands of DNA. The series of 4 distinct nucleotides along the backbone of the DNA, read 3 at a time, provide a blueprint to tell the body how to arrange the 20 different types of amino acids into protein strands. Once formed, these strands fold into complex 3-dimensional structural, hormonal, or enzymatic protein molecules that make up all the tissues and chemical processes in our bodies. In cancer, the DNA is damaged by either substitution or deletion of some of the nucleotides along the strand, resulting in miscoding of the amino acid protein chain. The abnormal sequence interferes with the normal 3D folding of the molecule, which, depending on the damaged segment, can result in abnormal cellular function, including cancerous transformation. Targeted therapies block or accentuate various molecular steps in cancer growth and proliferation triggered by these mutations. For instance, signal transduction inhibitors like tyrosine kinase inhibitors or TKIs are chemicals that block signals passed from one molecule to another inside a cell. Blocking these signals from the abnormal proteins can affect many functions of the cell, including division and death. Gene expression modulators like triplex-forming oligonucleotides or TFOs bind in the major groove of duplex DNA and can suppress the mutated gene from actually producing its abnormal protein. Apoptosis inducers like apoptosis activator 2 promote natural programmed cellular senescence and death or apoptosis. Some cancers have strategies to avoid apoptosis, with extreme cellular examples attaining an immortal status. Apoptosis inducers overcome the cancer's mortal resistance. Angiogenesis inhibitors like sorafenib block the tumor's ability to recruit new blood vessels, thus limiting the available energy supply and suppressing tumoral growth. The next major category of cancer treatments are hormonal therapies. Some tumors like breast, ovarian, and prostate grow and proliferate in response to the natural sex hormones in the body. Hormonal therapies like tamoxifen and flutamide block these hormones and restrict tumor growth. Radiation therapy is another tool in our armamentarium of cancer treatments. Remember, cancers are essentially caused by mutations in our DNA. If we can selectively destroy the abnormal tumoral DNA, we can slow or stop cancer proliferation. Using a highly modulated beam of ionizing radiation, the tumor cells are exposed to a lethal dose of X-rays. The beam is shaped to conform to the contours of the cancer mass and is administered in multiple projections to limit damage to the overlying tissues. The cumulative radiation exposure from the multiple projections is sufficient to selectively destroy the DNA of the cancer, preventing tumoral reproduction with minimal injury to the surrounding normal tissues. Immunotherapy uses our own immune system to help fight cancers. Some therapies help us train the immune system to recognize the tumor cell, sort of like a vaccine for cancer.
This one's good. This one's bad. Okay, this one is still bad, but he's trying to trick you. Whoa.
Others boost and disinhibit the immune system to attack certain DNA-damaged cells in the body. The supercharged and disinhibited immune system can then attack cancers like melanoma that possess strategies to avoid both detection and destruction. A revolutionary new therapeutic immune process actually genetically engineers our own T cells to recognize certain cancer-specific proteins, allowing them to seek out and destroy these elusive tumoral elements. Known as chimeric antigen receptor T cells, or CAR-T immunotherapy, the process starts by harvesting
What's this?
and separating our own T cells from the bloodstream. Outside the body, the cells are kept alive in a culture medium. Through various methodologies, including a virus vector, a new strand of DNA that encodes a cancer-specific protein receptor is inserted into the T cells.
Ow.
Once inserted, new receptors appear on the harvested T cell surface. This new T cell is then incubated in the culture medium, where it divides into a genetically identical colony. The cultured and modified cells are then harvested and injected back into the body. While the cancer eludes detection from the native T cells, the new receptor protein recognizes specific antigens on the cancer cell surface, enabling our own immune system to defend our bodies against the tumor. Most of the time, your team of oncologic doctors will use a multi-therapy approach to treat a particular cancer and maximize survival. With this summary, I want to take the last few minutes to review a couple of interesting theories regarding cancer prevention and therapy, including the role of diet. As evident by the avid tumor uptake of the glucose analog FDG, we know aggressive cancers require a lot of glucose to support their ramped-up metabolism. Therefore, it would stand to reason that limiting these cells' access to their preferred energy source may help with both cancer treatment and prevention. Dr. Walter Longo is an Italian-American biogerontologist, cell biologist, and author of The Longevity Diet. Based on over 25 years of research and innumerable animal studies, Longo advocates intermittent fasting and carbohydrate restriction to reduce the risk of many senescent disease processes such as heart attack, stroke, Parkinson's, Alzheimer's, and possibly even some cancers. Remember, when a cellular mutation occurs that creates a cancerous lesion, the nascent tumor is vulnerable. Before it recruits its own blood supply, the early cancer is highly dependent on the local cellular environment to supply its requisite energy and proteins necessary for cellular reproduction. An intermittent fast may literally starve these energy-dependent cells, pushing them towards senescence and apoptosis. Basically, the fast gives the body a chance to rid itself of bad cells and do a little house cleaning. In addition, the intermittent fast may actually boost the efficacy of cancer therapy. Chemotherapeutic agents preferentially damage the rapidly dividing cancer cells. To repair the damage, the cancer requires a steady supply of glucose for energy and protein for structural repair. Limiting protein and carbohydrate intake during the weeks of chemotherapy administration theoretically may kill more of the tumor than chemo alone, basically a synergistic effect. This would be followed by a high-protein, high-carbohydrate diet to allow repair of the normal tissues after treatment. Dr. James DeGregori and Robert Gattenby presented an article in the August 2019 issue of Scientific American entitled Darwin's Cancer Fix. We're all familiar with Charles Darwin, the 19th century English naturalist and father of the modern evolutionary theory, whose now famous finches graphically depict the evolutionary pressures responsible for the island-specific unique beak adaptations that are found on these closely related Galapagos species. The authors propose a Darwinian microenvironment that may actually impact cancer growth and treatment. As we know, all the cells of our body, even cancer cells, require energy, protein, and oxygen to survive. According to DeGregori and Gattenby, just like Darwin's finches, all of these cells compete for these local resources, and the regional microenvironment may actually favor one cell type over the other. For instance, obesity creates a systemic inflammatory state that may favor the growth of certain tumors in the cellular milieu, such as breast, liver, kidney, colon, and GYN cancers. In fact, in women with stages 1 through 3 breast cancer, data suggests that weight loss and exercise may significantly reduce the risk of recurrence in this particular patient population. On the treatment side, the tumor mass consists of a variety of cancer cells, the majority of which will respond well to traditional chemotherapeutic agents. A few cells, however, may possess genetic mutations that make them resistant to therapy. If the maximum tolerated dose of chemotherapy is administered, all of the sensitive cells die out, leaving only the resistant or most virulent cells. With no local competition for protein and energy, these cells then grow unchecked, resulting in a recurrence of a more aggressive, untreatable form of the original cancer. However, De Gregori and Gattenby suggest that if we only administer the maximum effective dose of chemo, the tumor mass will shrink, but some of the sensitive cells remain and compete with the resistant cells, preventing them from growing out of control. When the tumor does grow, an additional effective dose of chemotherapy is administered and the cycle continues. As a nod to the Darwinian model, this treatment regimen is described by the authors as adaptive therapy, basically allowing the patient to live with but not die from their cancer. Keep in mind, as for now, both the longevity diet and adaptive therapy for cancer treatment are theoretical and will need controlled patient studies to prove efficacy. As such, neither one is currently recommended without the direct supervision of your oncologic physician. As you can see, our current knowledge of cancer development and treatment is quite extensive, but we still have a long way to go. In the end, we may just prove scientifically what your grandma knew all along. To stay healthy and potentially reduce your risk of cancer, eat right, and strengthen your immune system with regular exercise and a good night's sleep. Thanks for watching.